attendees in as well. So here we go.
Great.
Go ahead.
Amazing. 1 second. Let's see. Do we want to wait for people to trickle in? I think people are going now. So I might just wait another couple minutes as people come in. I think the attendees are coming in now. And while we wait, I see, Don, the Q&A bucket. I'm not sure if I have the option to ask questions, but do the attendees have the option to ask questions? I guess that's
They do.
Great.
They do. Yeah. And as just the one quick thing to mention as well for anybody that's watching on Zoom, you're getting a pretty quick response overall. So Stephen and Valerie, whenever you do start to ask questions, just keep in mind that there is a delay between us and then LinkedIn, about 30 seconds or so. So, there will be a little bit of a delay.
Great. I think I'll just wait another minute or so as people trickle in. But also, Don, as you said, these clips and things will be posted, so that'll be for people who missed the intro. What I say won't be missing much. So as long as they hear our fantastic panelists, that would be great. Cool. Okay, well, I think I'll kick it off. And so as people come in, hopefully they join for the important pieces. But hello and welcome everyone. My name is Stephen Tan, the CEO of Woo Consulting Group. We are a biometrics FSP and kicking off our educational webinar series in celebration of 30 years of doing business with pharma and biotech. Today, we are joined by our friends, the Harvard T.H. Chan School of Public Health. As a fellow alum of Harvard College and the Harvard Business School, I'm thrilled to introduce our moderator for today, Valerie Finnemeyer. Valerie is a manager of programmer development at the Center for Biostatistics and AIDS Research at the Harvard T.H. Chan School of Public Health. She coordinates study programming projects for the statistical programming team at CBAR and develops macros and other tools to improve efficiency. She received a master's in leadership from Northeastern University and a PhD in chemical physics from Kent State University. The floor is yours, Valerie.
Thanks, Stephen. All right, well, welcome to everyone. Today, we're talking about accelerating clinical trial development, case studies in adaptive trial design and timeline optimization. We're going to be hearing from 3 researchers as they present case studies across different disease areas and clinical trial phases. Now, because myself and 2 of the speakers are from Harvard, like Stephen said, I'm going to do a quick 30-second intro to a little bit of a history lesson there. So the AIDS Clinical Trials Group, or ACTG, established their Statistical and Data Analysis Center within the Department of Biostatistics at Harvard, at their School of Public Health way back in 1989. This was then transformed in 1995 into the Center for Biostatistics and AIDS Research, or CBAR, to facilitate broader statistical leadership in AIDS clinical trials, and has been expanded over its 35 years of expertise to include other infectious disease research areas. And some of those are what we're going to be hearing about today. So I'll start then by introducing the 3 speakers we're going to be hearing about. First off is Mark Gigante. He's a research scientist at CBAR. He received his PhD in biostatistics from Vanderbilt University and has over 15 years of experience working as a biostatistician in HIV and infectious disease-related projects with a focus on study design considerations for clinical trials. He'll be talking about a phase 2/3 platform treatment for COVID-19. Our next researcher is Linda Harrison. She's also a research scientist at CBAR. She received her PhD in biostatistics from Harvard and has over 15 years experience working as a biostatistician on infectious disease clinical trials. Her current research focus is on multi-drug regimens to treat tuberculosis, and she'll be walking through a randomized adaptive dosing strategy for TB as she speaks to us today. And finally, we're also joined by Dr. Hongkong Wang, who's currently a tenured full professor of biostatistics at Georgetown University Medical Center. She received her PhD in statistics from the University of Rochester. Dr. Wang has been collaborating with physicians and medical researchers for over 20 years in clinical trial design and data analysis in cancer research, and will be talking to us about a phase 1 oncology design strategy. So with that, I'll pass it over into our first speaker, Mark Giganti, lead us off. I'm going to go ahead and share the slide.
While you're pulling that up, Valerie, thank you very much for that really nice introduction. And thank you to Wu Consulting for inviting me to be on this webinar panel. As Valerie indicated, today I will be presenting the ACTIV-2 study design, a phase 2/3 platform treatment trial for outpatients with COVID-19 that my colleagues and I helped design and implement. Next slide. So to really understand and appreciate the ACTIV-2 study design, I actually think it's helpful to jump in the proverbial time machine and go back to the summer of 2020. As you recall, this was early during the COVID-19 pandemic. And at this time, there was very little known about proven treatments for COVID-19. And at the same time, there was multiple candidate treatments that were in the pipeline that needed to be evaluated and investigated. And so it became very clear to my colleagues and I, as we started protocol development, that we needed to design a study that could rapidly and efficiently evaluate the safety and efficacy of these multiple investigational agents that were in the pipeline. And ultimately, we settled on a phase 2/3 platform trial design that, in my opinion, had 3 defining characteristics. First, the study was a randomized, blinded, controlled platform that allowed agents to be added and dropped during the study. Second, there was an allowance for a seamless transition from phase 2 to phase 3 evaluation for promising agents. And then finally, there was this pooled placebo component such that when 2 or more agents were being tested concurrently, it was possible to use the same placebo to evaluate both agents. Next slide. So to hopefully further highlight these key design characteristics, I thought it would be helpful to share this hypothetical study schema to illustrate these components more clearly. Using 3 agents to 3 hypothetical agents to consider and walk us through. And so the first thing I want to highlight on this slide is that the ACTIV-2 study had allowance for enrollment to be staggered, such that enrollment could start with a single agent, say agent A, and then as agent B became ready for evaluation, it could be added, and eventually agent C, such that enrollment could be occurring concurrently to all 3 agents. The second thing that I want to highlight is that all 3 agents, agent A, agent B, and agent C, start under phase 2 evaluation. However, as I mentioned on the previous slide, there was this allowance for a seamless transition from phase 2 evaluation to phase 3 evaluation, as illustrated by agent B. And then finally, I want to bring your attention to the single bar indicating placebo. And there's 2 things I want to highlight about this pooled placebo aspect of the study. First, there is a temporal restriction on this placebo, such that only participants enrolling into the placebo arm at the same time period when enrollment is open to an agent of interest would contribute to the placebo comparator arm for that agent. And then second, highlight that it was possible for a participant in the placebo arm to contribute to the evaluation of multiple different agents as part of the comparator arm. Slide. So I'm very happy to share that the ACTIV-2 study is complete and by all measures was a quite successful study, thanks in no small part due to the design characteristics which I've discussed on the previous slide. Notably, the use of a master protocol allowed the study team to evaluate 8 novel potential therapeutics over an 18-month period under a single protocol. There was one agent which did transition seamlessly from phase 2 to phase 3 evaluation. And for that agent, there was a 20% reduction in the total sample size that the study team needed to enroll compared to what would have needed to be enrolled if separate phase 2 and phase 3 trials were conducted separately. And then finally, in our setting, the use of a shared placebo led to a 26% reduction in the sample size when multiple agents were under evaluation. And so with that, I'll ask Valerie to briefly flash the references slide, and then I will turn it back over to her.
Thanks, Mark. So we are going to save audience questions until the end, but between speakers, I am going to ask a question or two before we move on to the next presentation. So Mark, you indicated a couple of times that there was a seamless transition between phase 2 and phase 3. Can you talk a little bit more about that? How much of a lag time was there between the phase 2 evaluation and start of the phase 3 enrollment period?
Yeah, that's a really good question, Valerie. So, it's important to acknowledge with any study that there is sort of a amount of time that needs to be allowed from when enrollment stops to when the results of the study are available. There needs to be allowances for data cleaning. There needs to be allowances for time for the statisticians to analyze the data and then share those results with, say, a DSMB in order to make a decision on how to proceed. And so in the interest of time, I didn't mention this, but now that we have time, I can highlight it. In this study, we actually built in 2 interim analyses during the phase 2 evaluation. With the first occurring when enrollment to phase 2 was halfway complete. And at that time, we would make the decision, once enrollment to phase 2 was complete, would it be reasonable to continue enrolling into phase 3 until the complete phase 2 evaluation was finished? And so that would allow us to continue enrolling into phase 3 without introducing any gaps once phase 2 enrollment completed. And then once phase 2 enrollment was complete, we would make a final determination as to whether we should continue enrolling in phase 3 or if we should halt the study at that time point. So essentially, in order to be seamless, we introduced an interim evaluation during phase 2. Hopefully that's clear.
Yeah. Thanks, Mark. And in interest of time, I think we'll move on to the next presentation. So I'll stick to the one question for now. So with that, I'll pass it over to Linda to talk about her TB study.
Great, thank you so much, Valerie, and for Woo Consulting for hosting this webinar. So I'm going to talk about another ACTG study. It's ACTG A5409, and it's also called the RAD-TB platform. It is a phase 2 randomized adaptive dose-ranging open-label trial of novel regimens for the treatment of pulmonary tuberculosis. And this study is ongoing, but the study design for wave 1 has been written up in Trials Journal. And if you scan that QR code at the top right, you should get to that publication if you're interested. So you can move to the next slide, Valerie. So there has been a recent resurgence of TB drug development. We went through a 40-year period where no new tuberculosis drugs were approved. And then in 2012, bedaquiline was approved. And since then, multiple new chemical entities have entered either late preclinical or early clinical development. So the hypothesis of this is that novel regimens evaluated by the RAD-TB platform will have superior early efficacy and acceptable safety and tolerability relative to the HRZE standard of care treatment for drug-susceptible pulmonary tuberculosis. You can move to the next slide, which should be the study design of wave 1 of this platform. So I'll spend a little bit of time on this because this is the main design of wave 1. So we're taking people with expert MTB positive drug susceptible pulmonary tuberculosis. So in sputum that they cough up from their lungs, we are able to detect TB by this point of care expert test. We take only adults in this protocol because it's very different measurements of TB cure between adults and children. Then in this first wave of this platform, we are randomizing patients to one of 6 arms, where we have arm 1, which is our standard of care arm, our HRZE combination therapy of 4 drugs for 8 weeks, followed by our HR extension from week 8 to week 26. That arm is going to have 90 participants. And then each of our experimental arms will have 45 participants. They will be given a novel intensive phase of a TB drug combination. In arm 2, we'll give BPAL, which is the bedaquiline protonamide linezolid combination of 3 drugs. This combination has been successful in drug-resistant TB and is being trialed here in drug-susceptible TB. The aim of this first wave of the platform is to determine which drug we should replace that linezolid with. So 2 drugs in the same class of linezolid are being trialed. In arm 3A and 3B, we are trialing TBI 223 at 2 different doses. And in arm 4A and 4B, we are trialing sutazolid at 2 different doses. The primary outcomes are for efficacy at week 6 and safety at week 8. But because TB has a long-term outcome and you really need to follow people for 6 months of treatment to see if they remain TB-free, we have follow-up right up to a year to assess the secondary efficacy durable TB cure outcome. So you can move to the next slide, Valerie. So what's going to happen in subsequent waves of this platform study? Well, the ultimate aim of RAD-TB is to find an optimized 4-drug regimen for drug-susceptible pulmonary tuberculosis. But we're trying to do a sort of rational combination regimen finding here, where we think bedaquiline and protominin would be a good backbone, but the linezolid drug may be safer. It may be a safer combination if we switch that linezolid drug to 2 other drugs in that combination. So we need to sort of find the dose of those drugs and give them in combination before moving forward. So a subsequent wave might take regimens that graduate from wave 1 and then add a 4th drug in order to be more efficacious and kill the TB bacteria more quickly. Alternatively, depending what drugs come through the pipeline, we may switch and do the second wave on other 3-drug regimens where we assess novel agents from different drug classes that weren't considered in wave 1. So this isn't a seamless design like the COVID study. This is using waves where we have time to analyze the data before moving on to the next wave. So it doesn't use an interim review committee to do that, and the team are involved in that. We do have a hard and fast graduation rule, which is based on safety, that if a regimen is shows a greater than 15% higher observed probability of grade 3 or higher adverse events through week 8 compared to the standard of care HRZE, we won't consider that regimen moving forward. But once a regimen passes that safety guideline, we'll assess its efficacy using the 6-week efficacy outcome based on time to TB positivity over 6 weeks. And then also we will look at the regimen's risk-benefit profile, the overall safety, as well as the PK parameters, because we're trialing those new drugs at 2 different doses. So I'll end there. Thank you very much.
Thanks, Linda. So you talked a little bit about the fact that you don't have the same type of interim review that was in the seamless design previously. Can you talk about what you did do for an interim review, and did you have interim efficacy or safety stopping guidelines that you were following?
Yeah, thank you, Valerie. So I actually forgot to mention that this study is ongoing. So currently it's recruited 24 participants in that first wave, and we're targeting 315 participants in that wave. So we do have interim monitoring. So the interim monitoring is largely based on safety. This is because there is a delay for efficacy data in TB. So we're basing our efficacy outcomes on TB cultures, and a TB culture takes 6 weeks to grow in the lab. So we won't have available efficacy data until really the end of the study, because it's a 6-week efficacy outcome, then 6 weeks to grow. So we're basing our interim reviews on safety, and really that graduation criteria about the 15% observed higher probability of grade 3 or higher adverse events at the end of the study. So in the middle of the study, when we have 50% of information on all the patients in the experimental arms, we're going to do a safety rule where if we're unlikely to meet that 15% at the end of the study, we're going to stop that arm early because of safety. And we're basing that on seeing a 30% higher rate at the interim in review, which sounds like a very large rate, higher than the standard of care. But at the interim review, bearing in mind you'll only have about 22 or 23 participants per each experimental arm. So it is quite an early look.
Yeah, it is a really small group of participants. How did you end up determining that sample size?
Yeah, so the sample size is based primarily on the efficacy, and they really want this trial to be a sort of phase 2A kind of study where they'll trial some drugs that haven't yet been given in combination in the patient. So it's kind of a very early phase study, similar to phase 1, to check the safety. But they would like an efficacy readout for this study in order to move to the later phase 2C studies that we have in TB and the phase 3 studies. So they wanted to get this early efficacy read, and that's so the sample size is based on that, and the time to positivity results in the liquid culture that we're actually getting at week 1, 2, 3, 4, and 6, and fitting a model on the time to positivity over the first 6 weeks to make the best use of our efficacy data.
Thanks, Linda. All right, with that, we will turn it over to our last speaker to talk about phase 1 oncology trial design. Hongkun.
Thank you, Valerie, for the nice introduction. And I'm very much honored to be here today with researchers from Howard Chan School of Public Health. And I would like to thank Stephen, the Wu Consulting, organized this event. So today I will basically talk something about the phase 1 oncology trial design based on my experience. So Valerie, can you move on to the next slide? Thank you. So we know for the phase 1 trial design, based on different disease area, methods, approaches will be all sort of, different. Really, depends on the disease area. For the phase 1 oncology trials, the conventional way is we would like to find the maximum tolerated dose level so that we could identify, the best dose that would treat patient and get, the ultimate efficacy. And to do that, we based on the finding the probability of the dose limiting toxicity, which will not exceed some pre-specified target. Traditionally, we usually aim at around 30%. We won't want to exceed that limit. And there have been various design methods in the phase 1 oncology trials, and particularly people would like to classify them into three categories. For example, the rule-based design, the 3+3. And then the model-based, for example, the continuous reassessment method. And then there's the model-assisted design, the MTPI or the bone design or the keyboard design. So because of these different methods, today I will just give an introduction from the 3+3 design to the bone design. Next slide, please. For the 3+3 design, it's a very straightforward design, and people have been using it for years until in the recent 10 or 20 years, there's the Bayesian method come to stage. For 3+3 design, you do not have to know any statistics. So basically, every cohort patient, 3 patients will enter to the lowest dose level, starting the dose level. Then you would observe based on the number of DLTs. If you observe 0 DLT, you escalate to the next higher dose level. You observe 1 DLT, you will enter 3 more patients at the same dose level. And if you observe 2 to 3 DLT at this, you would stop the trial dose escalation. And the MTD will be identified as a previous dose level. But if you have 1 DLT observed, then you would enter 3 more and observe if they're totally 1 out of 6 or 2 or more than 1 out of 6 DLT to decide whether you stop the trial or continue the trial. So this is very much easy implemented, and it's very popular among oncologists. So they do not have to each time come back to ask a statistician, should I do dose escalation? They can do it themselves. Next slide, please. Recent years, we have seen many methods to try to improve the performance of the 3+3. All the 3+3 is very straightforward. However, there's no associated statistical, properties. And on average, people did the simulation. It actually would identify the MTD on the targeted DLT level 25% instead of whatever the 30% we want. So recent years, there are all the new methods emerged. Particularly, I would like to talk about today is the bone design, is the Bayesian optimal interval design. This one has gained quite popular recent years, and it received the fit for purpose designation from the U.S. Food and Drug Administration back in 2021 as a tool for drug development. So I have seen many protocols, many designs use this bone design. This is a so-called model-assisted design. It combines both the predetermined algorithm to guide the dose escalation or de-escalation in the rule-based design, and then use the Bayesian statistical method to guide the dose escalation, de-escalation rule. Next slide, please. So you can see this flowchart of bone design is provided in the original designer, Dr. Yuan and others in MD Anderson Cancer Center. You start the patient at the lowest dose level. Then you treat the patient and you look at it, whether this patient has the DLT or not. And based on the number of DLT observed at each dose level, you have a pre-specified dose escalation, de-escalation boundary, the probability, the lambda E here or lambda D here. And then if the observed DLT rate is less than or equal to lambda E, then you would escalate dose level. But if it's beyond the lambda D, you would de-escalate the dose level. So you can see that it's still pretty straightforward, looks like the algorithm based, but the lambda D and the lambda E here incorporate the Bayesian statistical models here. So you have gained better statistical performance for this design. Next slide, please. For the bone design, you would have associated decision table. So comparable to a 3+3, right? Physicians would have this table to see, oh, okay, I have treated a number, for example, patients 5. Then I would escalate if I observed maximum 1 DLT. But I would stay in the current dose level if I observed 2 DLTs. And I would do dose de-escalation if I have observed more than 2 DLTs. So every bone design would associate with this kind of decision table. And the connection of it is bone design, you're not restricted to every dose level, maximum 6 patients, like in the 3+3 design. You can define number of patients you would like per dose level. And decision would be based on each dose level, the DLT observed. Next slide, please. Nowadays, because of the advance of all this biology work, and then people would like to find more information per specifically the dose level. Now the bone design migrated to the backfill bone design. People would like to see, okay, at a particular dose level, I would like to get more information about the safety and efficacy. Then the backfilling bone design seamlessly incorporate the backfilling cohort into the design and try to look at the specific indications and biomarker positive tumors. In these slides, you can see particularly dose level 2, after it is cleared for the safety issue, you can use this dose 2 as a backfill cohort. And similarly, if you escalate to dose level 3 and dose level 3 is cleared for the safety, then when to escalate to dose level 4, you can use dose level 4, dose level 3 as a backfilling cohort. Next slide, please. Here I give some reference for the original the bone design methods and what the FDA approved. And then there's a software. If you want to use this method, there's software from the MD Anderson website, free, easy to use. I'll stop here. Thank you.
Thank you very much. So you mentioned that the Bayesian optimized interval design has received the fit for purpose designation from the FDA in 2021. Are you aware of clinical trials that are leveraging this design at this point?
There are many. So yeah, so disease area from, let's see, the latest stage small cell lung cancer to non-Hodgkin lymphoma cancer. All these cancer areas, they try to use this the bone design, and also there's this targeted antibody drug conjugate therapies. They try to use this as well. Right now, I can see the bone design gain more and more popularity. On average, protocols studies I see, like almost half of them going to Georgetown use the bone design. Very popular now. Yeah.
Thank you very much. All right, I think we're at the open Q&A section. So I know in Zoom, at least we have the Q&A box for you to put your questions in. I'm not sure what the interface is on the LinkedIn side. But I do have a couple that have already come in. Mark, first question, just to clarify something that came up in your presentation, is the interim analysis per agent for your study?
Yeah, so with Active2 being a platform trial with 8 different agents, there was one study, one protocol, but each of those agents was evaluated separately. And so there was separate interim and final analysis for each of those 8 agents.
Thanks. And Linda, another question for you. What was the culminating trigger or triggers that spurred the resurgence of TB drug development after that 40-year drought that there was?
Yeah, I think this was really drug-resistant TB. So the sort of standard of care HRZE for drug-susceptible TB was really developed in the 60s and the 70s, and then it was rolled out across the world. And people primarily treated drug-susceptible TB as a very standard way across countries in their TB programs. And then some drug-resistant TB emerged, and in Peru, in particular, some doctors tried to see if they could treat it with other drugs. And then there were a series of cohort studies in Bangladesh. And from that, bedaquiline to treat drug-resistant TB emerged. And then the other drugs, as I alluded to in my presentation, really came from drug-resistant TB, and they are very promising. And now we can have oral treatment for drug-resistant TB. So now they're sort of being moved to see if they could kill the bacteria quicker for drug-susceptible TB, because that standard of care is 6 months in length. So the hope eventually is to reduce that length of treatment for drug-susceptible TB. So really what triggered it was this emergence of drug-resistant TB.
Thanks, Linda. So this is a question for all of our panelists. We're talking a lot here about all the amazing things, the advantages that some of these optimized trial designs can bring. What do you see as a drawback or a reason for concern as you're going into using one of these trial design approaches? Mark, you're nodding, so I'm hoping that means you're ready to answer, so I'll call on you first.
Yeah, absolutely. And, having seen this study, the Active-2 study from its design stage through its implementation and now all the way through its completion, it definitely is important to acknowledge that it isn't always a smooth sailing ship, right? One should not go into an adaptive study design and think that it will just go on without its flaws. Flaws isn't the right word. Without its choppy waters, to continue the sailing nautical theme. The challenge I'd like to highlight for those who are attending the webinar is it requires a lot of deliberateness in thinking through during the design phase what are important scenarios, what are important contingencies that you need to plan for, and sort of make sure that the study is designed tightly and correctly, so that when the study does open it in practice, you're prepared. To give one specific example, in our study, we had a pooled placebo, which with 8 total agents under evaluation, there was one time period when 5 different agents were under evaluation. And so making sure our infrastructure for randomizing participants to 5 different treatments and a placebo was something that we needed to build out of our existing infrastructure. And we were able to do it, and it was reasonable to do, but it would not have been something that we would needed to build out if we were just doing a traditional trial. And so again, just deliberateness in planning for these studies is quite critical.
Thanks, Mark. Hung Kwon, did you have anything to add? What about concerns on the Bayesian methods in oncology?
I think lots of what Mark just said, I agree with, because in the early phase trial design, really statisticians and physicians need to work together to understand, what is the goal in this trial and what, this drug or this compound, the behavior and all this PDPK, all these things, right? You can't blindly, I say, just use a bone design or just use a 3+3 design. I think also because of early phase trial design, there are lots of constraints, right? You can't just randomly, put a lot of patients in it. So I think we as a team need to really have a better understanding to get the, the best design for the trial. That's all I want to say. Thank you.
Thanks. And Linda, anything additional from your perspective?
So I think for my study, we have a great team who are very responsive and complement one another. Our chair is a pharmacometrician, so she understands a lot about TB drugs and then also the statistics. And then I hopefully bring in a statistical collaboration, and then we have two clinical vice chairs. And I think we've all worked pretty hard to get the study to come together. So I think that deliberate planning and working really in sync with your team, as Mark and Hunquin have highlighted, is really important. And like sort of thinking through the big picture and the major details. I have always done sort of 2 or 3 arm studies before, and I'm glad I've had that experience before moving to a sort of 6 arm platform design. So I think it's always good to work in lots of different studies and sort of use your experience from one study to the next. Yeah.
Thank you very much. Kwon, coming back to you, we talked a little bit during Linda's presentation about sample size, and you mentioned that in the Bayesian method that you can sort of select how many participants you want at the different levels. Is there a method for optimizing how many participants you pull in?
I think typically per dose level, you can put a restraint, let's say 9 patients or 12 patients, right? You don't want to spend too many sample size, particularly for one dose level. So that's why in the bone design, we have built in, the stopping rule. If you exceeding this number of subjects, we should stop here. Or the total sample size for the whole study should be kept at what level. For the oncology study, particularly phase 1, early trial, we really have to pay attention to the sample size restraint. We really do not have that luxury to put hundreds of patients in it. So I think the recommendation for it, you can have 9 per dose level, 12 or 14, 15. All this is based on the per dose level, the maximum, the DLT level you can have for that. Thank you.
Thanks. And Mark, I'm going to come back to you for this last one. What were the actual graduation requirements for the transition from phase 2 to phase 3?
Yeah, so I began my presentation by, jumping us into a proverbial time machine back to 2020. And so to answer your question, we'll jump back into that time machine. So when we were developing this study, COVID-19 was an emergent disease. And very little was known about COVID-19. Typically, for a phase 2 study that you're evaluating, you will choose surrogate markers that are predictive of your outcome of interest in order to evaluate your therapeutic. For COVID-19, we didn't have that. And so the study team, after discussions, drew upon their experiences with other respiratory infectious disease studies, most notably influenza, and came up with a total of 6 different graduation guidelines to inform that transition from phase 2 to phase 3. 3 of them were based upon viral load. 1 was based upon symptoms. 1 was based upon pulse oximetry. And then finally, we had the safety net of hospitalization or death, such that if we were seeing some signal of improvement in hospitalization or death, we wanted to make sure that we continued that therapeutic into the next phase.
Thank you. All right, I think we are at our time, so I'm going to pass that back to Stephen to close us out here.
Great. Yeah, well, thank you to everyone for joining on LinkedIn or on Zoom, and we'll definitely be passing around some information afterwards and summarizing what we just discussed. But I want to thank all the panelists from that Harvard Teach Chan School of Public Health, and Hong Quen, one of our consultants. So very much thankful for everybody's time to join on a Friday, and I will give back the time that we did have to all of you. So thank you so much for joining, and look forward to getting a chance to collaborate in the future.